My name is Lawrence Rosenberg, and this is the Alpha Human Podcast. Before we begin, it's important for me to state, as I always do, that I am not a doctor, nor am I a medical or healthcare professional. And the content of this podcast and any information, advice, opinions, statements within it are definitely not intended and are not to be used as a substitute for professional medical advice, for diagnosis, or for treatment. You shouldn't disregard or delay in obtaining medical advice for any medical condition you may have, and you should seek the assistance of a healthcare professional for any such conditions, and always inform your doctor of any changes that you make or are thinking about making to your lifestyle. Okay, so in our last episode, we talked about how chromophobe cancer, the cancer that I happen to be afflicted with, and a few other cancers do not use traditional pathways to gain access to their two primary fuels, which are glucose and glutamine. In fact, chromophobe cancer, even though we do see the Warburg effect and glycolysis taking over for oxidative phosphorylation to generate energy, or ATP, given that there is clear mitochondrial dysregulation, glucose metabolism in these cancer cells is actually lower when compared to normal kidney cells. In fact, fatty acid metabolism, amino acid metabolism is also downregulated in this cancer. And so we talked about how chromophobe cancer and a few other cancers either naturally, in the case of chromophobe cancer, it's just the pathway they like to choose, or when forced to, maybe as in other cancers, if they happen to be faced with low nutrient availability, will turn to cannibalism to get their substrates for energy. They will consume the debris of broken and dying cells via autophagy and the lysosomal pathway. And by the way, we're not going to get back into that too much now because we did a whole episode on that last time, and I invite you to listen to that episode to learn more about this particular pathway with respect to how a lot of cancer cells, when starved off from their normal sources of nutrition, will turn to, again, cannibalism in order to get those building blocks and to get energy. But anyway, in the last episode, we also left off on a bit of a cliffhanger. I alluded to another very interesting facet of chromophobe renal cell carcinoma, and how it and a number of other cancers use a particular micronutrient to develop, survive, and proliferate, and how this micronutrient may also provide the very key to its destruction. So let me just say this, I'm saying micronutrient. You have macronutrients like fats, sugars, and proteins, or broken down for cancer cells and other cells, they turn into fatty acids, right? Glucose, or molecules of amino acids, right? So the cells use those broken down macronutrients for energy and to build or maintain their structure, while micronutrients, such as vitamins and minerals, act as cofactors, facilitating enzymatic reactions and playing key roles in various cellular processes. So you get your macronutrients that the cells use, and you get your micronutrients. And there are non-essential and essential micronutrients, right? So essential micronutrients are those that the body requires for normal physiological function and must be obtained through the diet, since the body cannot synthesize them in sufficient amounts. Non-essential micronutrients, on the other hand, are those that the body can produce in adequate amounts or are not vital for basic physiologic functions. And it turns out that an essential micronutrient that we all know as iron plays a crucial role in the production of hemoglobin, right? So a protein found in red blood cells that carries oxygen from the lungs to tissues throughout the body, and that also has a vital part in the proper functioning of enzymes involved in energy production, DNA synthesis, and immune system function, also appears to be a key factor in the progression of certain cancers. In fact, iron accumulation is typical of renal cell carcinoma or kidney cancer. And according to the paper, iron accumulation typifies renal cell carcinoma or tumor genesis, but abates with pathological progression, sarcomatoid dedifferentiation, and metastasis. I'm going to quote, "The current study investigates tissue iron levels in patients with renal cell carcinoma based on mounting evidence of a unique role for iron accumulation in this cancer type. Renal cell carcinoma diagnosis is significantly increased in iron and steel occupations." How about that? So those that work their lives in steel or iron manufacture tend to have significantly increased RCC or renal cell carcinoma diagnoses. And certain medical conditions associated with renal iron accumulation are also seen in those iron and steel occupations, and individuals with a polymorphism in the TFRC iron importer gene. Our findings indicate that renal cell carcinoma accumulates higher iron in the cancer cell compartment and tumor microenvironment relative to other solid cancer types or a non-neoplastic kidney, but that this iron accumulation intriguingly declines with renal cell carcinoma pathological progression to metastasis. So the a ton of iron being accumulated in these cells initially, but as that cancer metastasizes and spreads, it turns out that iron accumulation tends to decline. Okay, so this is really, really interesting. You know, I think the question is, why the hell does iron accumulate so profoundly in certain cancers like kidney cancer? Well, according to a paper entitled "The Disturbed Iron Phenotype of Tumor Cells and Macrophages in Renal Cell Carcinoma Influences Tumor Growth," I'm going to quote, "Iron is an essential nutrient that plays a complex role in cancer biology. It's essential for basic cellular processes such as mitochondrial respiration, and the enhanced metabolic turnover under cancerous conditions is controlled by iron-sulfur cluster proteins. Cancer cells evolved specialized mechanisms for iron acquisition, storage, and mobilization in order to ensure their enhanced metabolic turnover. That's why you see the disturbed iron phenotype in tumor cells. That's why you see increased iron accumulation in certain cancers." And according to another paper, by the way, entitled "Altered Iron Metabolism and Impact in Cancer Biology, Metastasis, and Immunology," published in the journal Frontiers in Oncology, iron metabolism must be tightly controlled within cells, and while fundamental to many cellular processes and required for cell survival, excess labile iron is toxic to cells. Increased iron metabolism is associated with malignant transformation of a cell. All right, so increased iron metabolism can cause a normal cell to turn into a cancer cell. It's also related to cancer progression, drug resistance, and immune evasion, some of the hallmarks of cancer. Also, iron bioavailability is rate limiting during DNA synthesis, and cells that undergo rapid division simply require more iron. It is therefore not surprising that iron accumulation is often observed in tumor tissues. And because tumors are typically low oxygen environments, cancer cells will adapt to hypoxic conditions by regulating iron metabolism compared to healthy cells. In fact, hypoxic cancer cells require higher levels of iron to maintain their aggressive proliferation and redox homeostasis. And that's according to the paper, "Iron Metabolism, State of the Art in Hypoxic Cancer Cell Biology." So back to kidney cancer, it turns out that according to the paper, "The Disturbed Iron Phenotype of Tumor Cells and Macrophages in Renal Cell Carcinoma Influences Tumor Growth," which I think we quoted before, the incidence of renal cell carcinoma, the incidence of its development was enhanced by iron overload in the kidneys of anemic patients who required chronic transfusion. So how about that? This is really interesting, right? They see incidence of renal cell carcinoma enhanced by anemic patients who require chronic transfusions, right? And by the way, repeated injections of iron, renal cell carcinoma development with increased metastasis to the lungs and lymph nodes in experimental models. So the more iron you pump in, right, the more toxic it becomes to the cell, and the more these cells end up turning into cancer cells. As I probably explained in the last episode, they do that to escape death. We'll probably talk about that a little bit more in a moment. But ultimately, when it comes to chromophobe cancer, right, so the cancer I'm dealing with, high amounts of iron deposits have been observed, and mostly within the tumor stroma. And the stroma, I think I explained in the last episode, that's the structural components that hold tumor tissues together, okay? Another interesting fact about elevated intracellular iron concentrations is that it also leads to increases in reactive oxygen species. And this is where you see these cells becoming malignant, okay? So what happens is increased intracellular iron concentrations in these cancer cells, right, which is needed for that increased DNA synthesis and metabolic turnover, leads to an increase in reactive oxygen species, right, a very potent free radical, which we explained all about in our last episode, causes lipid peroxidation, right, which if you recall in the last episode when we were discussing these free radicals, is the process in which reactive oxygen species will steal electrons from the lipids in the cell membrane, resulting in cellular damage and death. And of course, then those cells will die, or they will figure out how to survive by turning to glycolysis instead of oxidative phosphorylation to get their energy. And when they make that metabolic switch to survive, they end up becoming cancer cells in many cases. So in the previous episode, we mostly focused on that negative impact and disease-inducing aspect of reactive oxygen species. However, it's crucial to note that these free radicals also play a very important and beneficial role in many physiological processes, including controlling the flow of blood through our arteries, right? Killing pathogens such as bacteria and viruses, and regulating cell growth. And to that point, reactive oxygen species plays a dual role in cancer. Quoting from a paper entitled The Two Faces of Reactive Oxygen Species in Cancer, on the one hand, reactive oxygen species can promote proto-tumorigenic signaling, facilitating cancer cell proliferation, survival, as I was saying just a moment ago, and also adaptation to hypoxia. On the other hand, reactive oxygen species can promote anti-tumorigenic signaling, right? So it can be anti-cancerous, and it can trigger the oxidative stress that those cells need in order to essentially commit suicide and die, right? So, if they get that signal properly, and in most cases, they don't figure out how to escape death by turning to glycolysis, then they kill themselves, and the body gets rid of those potentially damaging cells that can cause things like cancer. And you know what? That's exactly what should happen to these damn chromophobe cancer cells that I've got. And in fact, due to a recently discovered form of programmed cell death known as ferroptosis, this is exactly the fate, or what should be the fate, of cells that end up becoming chromophobe cancer cells, right? So ferroptosis occurs as a result of increased reactive oxygen species levels due to elevated intracellular iron. Ferroptosis as opposed to apoptosis, right? Ferro as in iron, right? So you get these elevated levels of iron for reasons we just described, which lead to increases in reactive oxygen species, which should then cause ferroptosis, right? So that intracellular iron concentration should happen, And you should see a depletion of the, accordingly, you should see a depletion of the antioxidant, glutathione, that should then cause lipid peroxidation, right? So those membranes around those cancer cells should start to get damaged and die, and all good. That should be it. And again, that's according to a paper entitled "Reactive Oxygen Species Induced Lipid Peroxidation in Apoptosis, Autophagy, and Ferroptosis." Now, I've mentioned this before, but apoptosis and autophagy are forms of programmed cell suicide, and it's essential for many biological processes, including tissue development and turnover, immune response, and the maintenance of cellular homeostasis. In fact, a lot of people try to induce autophagy on themselves by having prolonged fasts. I also explained last episode why that could be bad, right? If you have cancer, in certain cancers that depend on autophagy in order to cannibalize other cells, or to cannibalize broken and damaged cells in order to get their nutrition, increased autophagy could be a bad thing. But nevertheless, if you don't have autophagy, and get rid of these abnormal cells, on the other hand, that could lead to the development of diseases such as cancer. And also, it can lead to ferroptosis, right? So removing damaged or abnormal cells is an important thing. And it's like a lot of things in cancer. Like reactive oxygen species itself, it can be good, it can be bad. Autophagy can be good, it could be bad. And so ferroptosis is really a recently discovered form of cellular death. Again, another form of programmed cell suicide like apoptosis and autophagy. And there's this paper, basically, again, that I've kind of been quoting from just now in talking about how autophagy and apoptosis is essential as a biological process, and it's important for removing damaged or abnormal cells that could lead to the development of certain diseases like cancer. And again, ferroptosis is one of these types of programmed cell suicides that has gradually become accepted as an adaptive feature to eliminate malignant cells, and plays a pivotal role in the depression of tumorigenesis by removing the cells that are deficient in key nutrients in the environment or damaged by infection or ambient stress. Again, according to a paper entitled "Ferroptosis, a New Form of Cell Death, Opportunities and Challenges in Cancer." Now, although ferroptosis primarily functions as a cell death mechanism to eliminate problematic or damaged cells, under certain circumstances, dysregulation or excessive activation of ferroptosis can contribute to disease pathogenesis or exacerbate existing conditions such as neurodegenerative diseases, right? So, for instance, you want ferroptosis, you want iron-mediated cell death, meaning when iron accumulation happens in these cells, you want the increased reactive oxygen species, you want the increased lipid peroxidation so that it just naturally gets rid of these cells and keeps them from becoming cancerous. However, in healthy cells, if this is happening, and you're having this iron overload, and you're having this cellular death, it could cause neurodegenerative diseases, right? So ferroptosis has been implicated in neurodegenerative disorders such as Alzheimer's disease, Parkinson's disease, Huntington's disease, as dysregulation of iron metabolism, oxidative stress contribute to neuronal cellular malfunction and ultimately the cell's demise. Ferroptosis can also lead to ischemia and reperfusion injury. So during episodes of ischemia, which is restricted blood supply and a subsequent reperfusion, which is the restoration of blood flow, oxidative stress and iron accumulation can lead to tissue damage and cell death via ferroptosis. So this, again, is a real issue that we have to look out for, which is why antioxidants are so important. Antioxidants are important in order to clean up the damage being caused, by these, this overload of iron. It's meant to clean and mop all this up. It's supposed to act as a mechanism to protect various organ systems, including the heart, the brain, and kidneys. When you end up seeing issues like ischemia and reperfusion injury. But left unchecked, that's exactly what you're going to get, right? You're going to get ischemia, you're going to get reperfusion injury, but you're also going to see, as a result of nephrotoxic drug exposure or sepsis, you're going to see ferroptosis at play, right? So excessive lipid peroxidation and iron-mediated oxidative stress plays a role in liver cell injury and inflammation as well. And as such, ferroptosis has been linked to liver disease, such as non-alcoholic fatty liver disease, NAFLD, and alcoholic liver disease, ALD. So, you're going to see a lot of damage to the body from unregulated, unchecked ferroptosis, right? And that's something you don't want. So there's always this healthy balance of you end up getting this increased liver accumulation due to stress or injury to the body, right? Like in ischemia, like in fatty liver disease, and you'll start to see this ferroptosis happening. And so what you're going to want to protect the body from, to neurodegenerative disorders, to non-alcoholic fatty liver disease, to cell injury due to inflammation, what you're going to want is you're going to want to see a healthy level of antioxidants to keep ferroptosis in check. So that starts to lead us down the path to, well, wait a second, what's actually occurring in chromophobe renal cell carcinoma, right? Again, the cancer that I'm dealing with. Since we're seeing a ton of iron accumulation in those cells, again, which is helping metabolic turnover and DNA synthesis and replication and proliferation of these cancer cells, right? Like, why isn't the reaction cellular death in chromophobe cancer? Why is chromophobe cancer cells being allowed by the body to continue to proliferate as it does with other cancers? Why isn't ferroptosis doing its job, right? You don't want ferroptosis in instances where it's playing a big role in neurodegenerative disease, right? But I kind of gave you the clue when I was explaining what protects healthy neurons and healthy liver cells from ferroptosis, right? I gave you a clue. Why isn't chromophobe cancer cells dying? Right? They're not dying for the same reason your healthy cells don't die from ferroptosis, which is what they're supposed to be protected, and that's because the mechanism to survive the excessive reactive oxygen species production taking place in the afflicted chromophobe cancer cells, the mechanism that's protecting it is the very mechanism that protects other cells like neurons from being damaged or dying and causing neurodegenerative diseases. What's protecting the damn chromophobe cancer cells is that there appears to be no depletion of the antioxidant glutathione, right? In fact, what you'll find is quite the opposite, right? Here's a quote for you. Chromophobe cancer cell tumors are characterized by glutathione, and glutathione again is an antioxidant, glutathione levels that are strikingly elevated and are 50 to 100-fold higher than normal kidney cells. And that's according to a paper entitled Hypersensitivity to Ferroptosis in Chromophobe Renal Cell Cancer is Mediated by a Glutathione Metabolic Dependency. And since glutathione is an important reactive oxygen species scavenger, balancing out the stealing of electrons that those free radicals are running around, running amok in those cells, causing a chain reaction where they continue to steal more electrons and damage the lipid membranes of the cells, of those cancer cells, glutathione comes to the damn rescue. The increased glutathione levels and GSSG ratios in chromophobe cancer can thus be considered as the main strategy for the tumor to overcome reactive oxygen stress originating from a dysregulated respiratory chain. According to the paper, Metabolic Reprogramming and Elevation of Glutathione in Chromophobe Renal Cell Carcinoma. Again, chromophobe kidney cancer is a case study in the Warburg effect. And we talked about the Warburg effect in the last episode, where these cells have damaged mitochondria, and in order to adapt, they switch over from the normal type of metabolism that they use to produce energy, to produce adenosine triphosphate, ATP, in order to survive, right? In order to produce energy, they move towards the fermentation of sugar, not because they're genetically programmed to get cancerous, but because there's been damage to the cell's mitochondria, and that turns on this metabolic switch, which causes these cells to become malignant. And again, we talked about Thomas Seyfried and his theory of cancer, and also why the genetic theory of cancer, and alternative theories are proliferating right now, and why it's highly likely that cancer in most cases is not a genetic disease, but a disease of metabolic dysfunction. Chromophobe cancer is a perfect case study. In 40% of chromophobe cancers, they see no mutation driving the cancer, and that is purely down to metabolic dysregulation or damage to the mitochondria. And so who comes along to the rescue of these cells that are trying to survive and accumulating tons of iron? And instead of dying, what they're doing is they're sucking up a ton of glutathione. They're turning it over, huge metabolic turnover of glutathione in order to survive reactive oxygen species damaging the cancer cell membrane. So these highly adaptable, sneaky little cunning cancer cells have yet again figured out a way to survive when they should simply die off. One of the hallmarks of cancer is their ability to acquire and develop mechanisms that allow them to adapt and survive in challenging environments. And with that said, it's often observed that the same mechanisms that contribute to the development and progression of cancer can also present vulnerabilities targeted for therapeutic intervention. And here's where the damn cancer cell that's using glutathione to survive also presents us with an Achilles heel, right? So look, if you think about it, if you can block glutathione in certain cancers where iron accumulation predominates, then the excess iron in the tumor cells would turn out to be a good thing, right? Ferroptosis would turn out to be a good thing. And, from a paper entitled Iron Accumulation Typifies Renal Cell Carcinoma Tumorigenesis, but Abates with Pathological Progression. I think we've quoted it before. Quote, the conventional understanding has long been that high iron is selectively advantageous to cancers. However, recent discoveries challenge the simplicity of this paradigm and suggest that increased iron levels may also suppress cancer through a regulated cell death process known as ferroptosis. Ferroptosis, which is morphologically and molecularly distinct from apoptosis, utilizes iron-dependent oxidative stress to induce cell suicide in response to reactive oxygen species levels that overwhelm cellular antioxidant defenses. Oxidative stress that suppresses transformed cell properties may become a liability for tumor progression and metastasis, and resistance to iron-dependent oxidative stress may therefore be necessary for cancer progression. So, couldn't have said it better myself. It stands to reason then, guys, that if there were a way to block glutathione, then chromophobe cancer cells should succumb to lipid peroxide-induced death. Right? I mean, sounds logical. Well, in a study led by researchers at Brigham and Women's Hospital, they successfully induced ferroptosis in chromophobe cancer cells via cysteine deprivation, and found that this may be an effective approach for treating chromophobe cancer cells. Excellent. Okay, that's a great study. And why cysteine deprivation? Cysteine is a another amino acid, and another one of those 20 amino acids. Well, according to the study at Brigham's and Women's Hospital, the results of which were published in a paper entitled Hypersensitivity to Ferroptosis and Chromophobe Renal Cell Carcinoma is mediated by a glutathione metabolic dependency. They say that, quote, cysteine, which, by the way, is, again, one of those really important 20 amino acids we mentioned in the previous podcast, is the rate-limiting substrate for the synthesis of glutathione. That's why you want to block cysteine. Cysteine uptake is mediated by the cysteine glutamate antiporter, XCT, aka otherwise known as system XC. That's the pathway. That's the pathway you want to inhibit. So rate limiting, by the way, I've used this term rate limiting. Rate limiting refers to the step or factor that controls or limits the overall rate or speed of a biological process. And in this case, the synthesis of glutathione, which means that the availability of cysteine is the factor that predominantly determines the rate at which glutathione is synthesized. In other words, when cysteine levels are limited, the synthesis of glutathione is restricted, and the overall rate of glutathione production is slowed down. This implies that cysteine is a critical and limiting component in the synthesis of glutathione, and its availability directly affects the efficiency and capability of glutathione production. And back to that paper, quote, here we report evidence that chromophobe cancer is highly sensitive to the induction of ferroptotic cell death via targeting the cysteine glutamate antiporter XCT. And CHRCC cells, chromophobe renal cell carcinoma cells, are dependent on cysteine uptake to preserve high glutathione content. We found that the markedly elevated levels of intracellular glutathione in chromophobe-derived cells are GGT1 dependent, thereby impacting sensitivity to reactive oxygen species, induced oxidative stress, as well as their proliferation in vivo. That means in living cells, right? In vitro means in the lab, right? Or in a petri dish. In vivo means in living cells. And so, by the way, GGT1, which I've just mentioned, is involved in the metabolism of glutathione. And so, quote, ferroptosis can be induced by inhibiting GPX4, glutathione peroxidase 4, which protects cells against membrane lipid peroxidation. Let me repeat that. Ferroptosis can be induced by inhibiting GPX4, which protects cells against membrane lipid peroxidation, or by limiting the supply of glutathione to GPX4. GPX4, by the way, is a key role in protecting cells against oxidative damage by neutralizing lipid peroxide, and it accomplishes this by using glutathione as a cofactor to reduce lipid hydroperoxides and protect cells and membranes against peroxidation. GPX4 is especially important for protecting cells against ferroptosis, right? So we want to block GPX4. And so if you've been listening to my podcasts on this subject, this is the fourth finding that I've been alluding to over the past few episodes. That had I done no research and had I not decided to deep dive into the medical literature to learn as much as I could about chromophobe cancer, then I would know none of this. Again, my initial oncologist simply told me in the space of just 2 minutes, there's no effective treatments for chromophobe cancer. That I need to stick with surgery if I can, because tyrosine kinase inhibitors, which is the choice therapeutic for most renal cell carcinomas, does not work in chromophobe cancer. And in the space of 2 minutes, that was it. That was although my initial oncologist appointment to discuss my options, was 40 minutes long, it ended up really right there in that crucial 2 minutes where they said, stick with surgery. There are no treatments, right? So had I not decided to dive deep into the literature, I would have never uncovered some of the things that I've uncovered, let alone this idea about killing chromophobe cancer cells via ferroptosis. And so, lo and behold, here's a study that shows us a potential pathway to treating chromophobe cancer cells via blocking glutathione via GPX4. This is why I cannot stress how crucial it is that all of you do your own research on whatever condition you may have. And beyond this important new information that I personally found, the added benefit of my investigation and the time that I've spent digging into the medical literature is that one of the authors of this paper on ferroptosis being a potential pathway for killing chromophobe cancer, one of the added benefits of the fact that it decided to really spend time studying the medical literature on chromophobe cancer is that one of the physician scientists on this study is now my oncologist, right? So I have a new oncologist who, by the way, is working in tandem with my other oncologist. But the point is, had I not done all of this digging, I would have never found this paper, and I would have never found this new form of potential cellular death that could wipe out chromophobe cancer cells, and I wouldn't have this new doctor. And so, I've had a bunch of episodes on chromophobe cancer, on cancer in general. And the reason that I'm doing this, the reason, that I've deviated from all the other episodes I've done, which are, lots and lots of interviews around enhanced human performance and mindset, as well as a number of enhanced physiologic, enhanced health episodes to help you, to help empower the human body and not just the human mind. The reason I've dived deep into the cancer rabbit hole here is, if anything, just so I can stress to all of you out there that it is crucial that you spend time investigating your own condition and that you don't simply just accept what you're told by any doctor, and that you get a second and third opinion and that you do your own investigation. Okay, nevertheless, back to the paper. Findings suggest that induction of ferroptosis via cysteine deprivation could be a highly effective therapeutic strategy for chromophobe cancer tumors, a tumor type for which effective treatments are urgently needed. Now, interestingly enough, while these studies suggest inhibiting cysteine as the pathway to reducing glutathione levels, Seyfried's press pulse protocol, which we discussed last time, right, where you starve the body of glucose through a ketogenic diet and where you starve the body of glutamine via certain drugs, and his hypothesis about, right, having this press and this pulse, right, this constant press of of the metabolism and this pulse protocol via the diet and the drug, right, via the starving of the starving of glucose via the diet and the blocking of glutamine, right, via the drug. The press is the ketogenic therapeutic diet, right, which you constantly maintain. That's the pressing of the system and the stress on the cancer cells. And then the pulse is that you'll have intermittent protocols of blocking glutamine via a drug, which will tip those cells into apoptosis, into cellular death. Anyway, the press post protocol. And interestingly, right, these studies suggest inhibiting cysteine as the path to reducing glutathione levels may also play a very effective role in the press pulse strategy, but not necessarily as Seyfried intended via blocking glutamine's availability as a fuel or blocking not necessarily as Seyfried intended via inhibiting glutamine's availability as a fuel or building block for cancer cell growth, but rather to provide an alternative pathway, or perhaps in conjunction with blocking GPX4, to killing these cells by inhibiting glutamine's conversion to glutathione in order to induce ferroptosis and allow reactive oxygen species free radicals to do the job they were meant to do. And in this instance, their job is to wipe out those chromophobe cancer cells. So, the way Seyfried, other people are thinking about blocking glutamine, and glutamine is important to the synthesis of glutathione, right? So if you block glutamine, right, yes, you could starve the cell of its building blocks, but You could also provide an avenue to ferroptosis, and right, and this is something that isn't necessarily what Seyfried was suggesting, or those involved in the press pulse hypothesis are thinking. But hell, this could provide two pathways to attacking the cancer cell, and we can find evidence of this strategy in the paper Glutamine Deprivation Induces Ferroptosis in Pancreatic Cancer Cells. And so I'll quote, glutamine deprivation inhibits pancreatic cancer cell growth and induces ferroptosis both in vitro and in vivo. So what drugs did they use to achieve this effect? Well, they tried two different compounds. Diazoxynorlucine, otherwise known as DON, D-O-N for short, and azacitidine, not in combination, but individually, to test the outcomes, and both seemed to work. As their conclusion was, quote, glutamine inhibitors, DON and azacitidine promotes reactive oxygen species formation and ferroptosis in pancreatic cancer cells. And that's what's really interesting, is that if we look again at Seyfried's press pulse strategy for managing cancer and the paper he wrote on this hypothesis, he also highlights diazoxynorlucine, not for its ability to induce ferroptosis, as in this other paper, but for its ability to, quote, deprive proliferating tumor cells of the glutamine needed for TCA cycle, anaplerosis, and synthesis of glutathione nucleotides and proteins. So when blocking glutamine, people, it turns out that we may be getting two different modes of action for killing these damn cancer cells. First, by ferroptosis, for those cancer cells susceptible to this type of cellular death, like chromophobe cancer, and second, via starving the cell of glutamine, one of the key building blocks besides glucose necessary for cancer cell growth and proliferation. However, one of the issues with diazoxynorlucine, or DON for short, is its toxicity. So let me read from a paper entitled, "We're Not DON Yet." That's pretty cheeky there. We're not DON yet. D-O-N, diazoxynorlucine. So what the paper says is the broadly active glutamine antagonist, 6-deazo-5-oxo-L-norlucine, otherwise known as DON for short, has been studied for 60 years. You hear that, people? 60 freaking years as a potential anti-cancer therapeutic. Clinical studies of DON in the 1950s using low daily doses suggested anti-tumor activity. But later phase 1 and phase 2 trials of DON given intermittently at high doses were hampered by dose-limiting nausea and vomiting. Further clinical development of DON was then abandoned. But recently, the recognition that multiple tumor types are glutamine dependent has renewed interest in metabolic inhibitors such as DON. To circumvent GI toxicity, prodrug strategies for DON have been developed to enhance delivery of active compound to tumor tissues, including the central nervous system. When these prodrugs are administered in a low daily dosing regimen appropriate for metabolic inhibition, they are robustly effective without significant toxicity. That, people, is very interesting, right? Imagine being able to kill cancer cells without resorting to harmful radiation and harmful chemotherapy, which ends up creating reactive oxygen species in order to kill the damn cancer cells. But we all know what an effect of lots and lots of reactive oxygen species are for the body. That's right, more damn cancer cells. So can you imagine if you could have a way of creating a non-cytotoxic effect on normal cells and targeting the cancer cell with a drug like DON, but having it altered so you don't have the tremendous vomiting and nausea and GI toxicity? Well, the good news is that there is a DON prodrug currently in clinical trials, according to the paper Discovery of DRP-104, a tumor-targeting metabolic inhibitor prodrug. I will quote, 6-diazo-5-oxo-L-norleucine, DON, is a glutamine antagonist that suppresses cancer cell metabolism, but concurrently enhances the metabolic fitness of tumor CD8 plus T cells. DON showed promising efficacy in clinical trials. However, its development was halted by dose-limiting gastrointestinal toxicities. But given its clinical potential, we designed DON peptide prodrugs and found that DRP-104 was preferentially bioactivated to DON in tumor, while bioinactivated to an inert metabolite in GI tissues. DRP-104 represents a first-in-class prodrug with differential metabolism in target versus toxicity tissue. DRP-104 is now in clinical trials under the FDA Fast Track designation. People, this is phenomenal. So that is really exciting news. I'm really excited to keep track of this particular peptide, this particular prodrug of DON, and see how it progresses. Nevertheless, we do have to get into what drugs are being used in the study that I mentioned, and in these other studies. There's lots of ferroptosis studies going on right now, and have been going on for the last few years. And also in the ferroptosis study that they did on chromophobe cancer, they didn't use DON, right? DON is the anti-glutamine drug, right? These other studies are using anti-cysteine pathway drugs. So what drugs are they using to induce only ferroptosis? Well, that, my friends, we will dive deep into in a next episode of the Alpha Human Podcast. Thank you for spending time with me today. Hopefully I didn't bore you too much with all of this jargon. But again, if I'm getting any point across to all of you, it is this. Again, victory through insight, right? That's the logo of the Alpha Human Podcast. Victory through insight. How do you gain insight? You do the research yourself. Don't be lazy, right? Study up on this shit. Get into this data. Get into your own condition and become an expert in it, so you could have viable discussions with your doctors and ask them the right questions, the hard questions that are going to help you understand your condition a heck of a lot better. And so with that, take care, and I'll see you soon.